Our last major category of important biomolecules are the nucleic acids. Now, maybe you've never heard of the nucleic acids before, but I know you've heard of molecules like DNA and RNA and ATP, and those are the molecules that fall into the nucleic acid category. So let's talk about them a little bit more in some more detail. Like the other groups of biomolecules, nucleic acids have monomers, dimers, and polymers. The monomers in this group are molecules known as nucleotides, and this is one nucleotide over here. If you put two of these guys together, you get the dimer, which is known as a dinucleotide. And if you put many of them together, you get the big molecules like DNA, and those are referred to as the polynucleotides. The monomers of nucleic acids are called nucleotides. Here we have a nucleotide molecule. There are three major parts to any nucleotide. In the center, it might surprise you to find that we actually have a carbohydrate molecule. It consists of 5 carbons. So here they are, 1, 2, 3, 4, and here's number 5 up here. So a 5-carbon sugar. Now attached to that 5-carbon sugar on one side will be a phosphate group, and on the other side is a molecule that we just generically call a base, or a nitrogen-containing base, or sometimes a nitrogenous base. So when you're looking for nucleotides, look for these 3 parts: sugar, phosphate, and base, all put together. Two different sugars can be found at the heart of a nucleotide: ribose and deoxyribose. Ribose is typically found in the nucleotides found in RNA. In fact, that's what the R of RNA stands for. Deoxyribose, on the other hand, is found in the nucleotides that make up DNA. That's what the D of DNA stands for. What's the difference in these two sugars? Well, to tell that, we really need to look at carbon number 2 here in the carbon ring, which I'm making red with a dot there. Notice that in the ribose sugar, there is an OH group attached to that carbon, a hydroxyl group. However, carbon number 2 in deoxyribose does not have a hydroxyl. Instead, it just has a plain old hydrogen. So because this sugar contains one fewer oxygens than ribose, we call it deoxyribose. And ribose has that extra oxygen. 5 different nitrogenous bases can be found in nucleotides. They fall in 2 categories. First, we have the pyrimidines. The pyrimidines are single-ringed structures, and they include cytosine, thymine, and uracil. Purines, on the other hand, are double-ringed structures. So they look kind of like a figure 8, and they include adenine and guanine. Now, how do you remember which ones fall in which categories? Well, there is a memory trick for that. I like to think of the word pyrimidine because it has a Y. And then if you look, the names of these two guys, cytosine and thymine, also have Ys. If you add a little tail to the U of uracil, you've got a Y there as well. So Ys, pyrimidines. Another way to remember it is that these single-ringed structures are kind of round, kind of like a pie. So pyrimidine. That's a silly memory trick, but whatever gets you there. Now, certain bases are found in certain polymers. Cytosine, thymine, adenine, and guanine are found inside DNA molecules. Cytosine, uracil, adenine, and guanine are found in RNA molecules. ATP is an extremely important nucleotide monomer. ATP stands for adenosine triphosphate. So let's look at its structure and see where it gets its name. Well, at its heart is a ribose sugar. Attached to that is an adenine, so that's where it gets the adenosine. And then if you notice, the phosphate group is really big. It has 3 phosphates attached, so that's where it gets the term triphosphate. ATP is the most important energy molecule found inside your cells. You can think of it as being like the energy currency or the energy money of a cell. In order to do certain activities like chemical reactions, you have to spend a little money, right? So in order to perform a certain chemical reaction, your cells have to break down a certain number of ATPs, and that provides the energy to perform the reaction. Now, you're constantly recycling ATP inside your cells, but scientists estimate that if you didn't recycle it, if you built it new every time, you'd have to build something like 90 pounds of this molecule every day just to stay alive. I just wanted to show you an example of some dinucleotides, the dimers of this group. Here's a dinucleotide. You can see that it is two separate nucleotides and that they are tied together through their phosphate groups. Now, dinucleotides are often used to shuttle energy from one place to another inside your cells, and we'll definitely see them in action in a later chapter. Examples of dinucleotides include NADH, FADH, and NADPH. So next let's talk about the big guys, the polynucleotides. Our first and most important polynucleotide is DNA. DNA stands for deoxyribonucleic acid. Yes, you do need to know what DNA stands for testing purposes. So what does the deoxyribo refer to? Well, you remember that refers to the type of sugar you find in the nucleotides. And nucleic acid just refers to the group in which it's classified. Now, what does DNA actually do? Well, it's sort of the master computer of the cell. It stores the instructions for running the cell and for determining what kind of cell it's going to be. Well, how does it do that? Well, the information that it actually contains allows the cell to manufacture the right RNAs and the right proteins at the right times. Remember that these molecules, RNA and especially proteins, do all the jobs of the cell. So the cell can control how it functions by turning the DNA on and off. And we'll get to how it does all of that stuff in a later chapter. Here we have the classic shape of the DNA molecule, known as the double helix. The double helix is shaped like a staircase. So these banisters, these handrails in this ladder, are made of the sugars and phosphate groups of the nucleotides. The steps in the middle are made of the bases, those A's, T's, C's, and G's, and this actually constitutes the genetic code. This diagram has flattened out a section of DNA so we can see how it's put together. If you look closely, you'll notice that this long strand is really just made of a bunch of nucleotides hooked together. There's one nucleotide, here's the next one above it, and so on. So it's a long string of nucleotides on both sides. Now notice what kind of bonds are holding together this sugar-phosphate backbone. They're solid lines, so they are covalent bonds, nice strong bonds. The backbones of DNA don't actually hold any of the code. It's actually the bases in the middle that hold the genetic code, so the A's, T's, C's, and G's here. Now, a couple of things to notice about these bases. First of all, the bases on one side are bonded to the bases on the other side of DNA, and they are held together with dotted lines. That's hydrogen bonds. The other thing to notice is that these nucleotide bases pair up in certain combinations. A's always pair up with T's, so adenines always pair up with thymines, and cytosines always pair up with guanines. So the rule is called Chargaff's rule, but we say A with T, C with G. They always pair up in the same pattern. We say that the two sides of the DNA helix are complementary to each other. In other words, they are not identical, but they match up in a specific predictable pattern. They complement one another. Your DNA is like a library that stores all of the information for running your cells and making them a particular type of cell. We would refer to that entire library as the genome. Now that library is going to contain books. Individual pieces of DNA, the books, we call chromosomes. If we were to open up a single book and look inside, we'd find the individual chapters inside that book, and they hold recipes for making specific proteins. We call these specific chapters genes. So a gene is a recipe for making one type of product. You put a bunch of those together, you get one book. That's the chromosome. You put all of those together, you get all the DNA. That's called the genome. We'll get into how your cell accesses all of this information and uses it later. Another important polynucleotide inside cells is good old RNA, or ribonucleic acid. RNA is a close chemical cousin to DNA. It uses the sugar ribose instead of deoxyribose, and it's put together a little differently too. Here you can see DNA and RNA next to each other so we can compare them. And what you can see is that RNA does consist of a long string of nucleotides put together, but it is a single strand, whereas DNA is a double strand. So it only has one strand of nucleotides and the bases just stick out into space. That gives it some different properties. Another important difference between DNA and RNA are in the bases that they use. DNA likes to use adenine, thymine, cytosine, and guanine, whereas RNA likes to use adenine, uracil, cytosine, and guanine. So they use slightly different bases inside their nucleotides. Now, RNA has a number of functions inside cells. The most important one is that it participates in the protein making process. So we'll see it in action in a later chapter. All right, so you're all done with chapter 5. Whew, that was a long one, huh? Make sure that your notes are thoroughly filled out and that you've completed all the year turns, and bring all of that to class with you so we can go over it and you can get a handle on all these fantastic molecules. We'll see you then.